CFS. Dr. Fiehn? So here's your pointer and your slide advance.
Thank you. Thank you. So we had a great introduction from the West Coast of the United States, San Diego, Bob Navio. It told us all about like how important metabolism is to understanding energy dysregulation, and talked also about systemic effects and the similarities between diseases that we should more focus on, and not just like one disease, but also the comorbidities, and he focused on the healing process. So that's a great prelude to what I want to say, because I have similar punchlines in a way. So first of all, let's have a look at metabolism. So that is what we study when we are undergrads, right? So we look at the major inputs and outputs in terms of energy metabolism, fats, proteins, glycolysis. And you see that they are linked. There's many ins and outs. It's much more, if you can actually look more closely, then there's all of a sudden lots of shunts, lots of exit routes, lots of more network effects than what you have studied when you were younger. So that while metabolism in this broad view is of course important, it's not the only one. So that is metabolism, what I show here, the metabolism of building and burning of energy and building macromolecules. But there's also a much more important, maybe, metabolism of regulation. So let's assume now that we have lots of data. And we have lots of data from humans. And then in aim 3 of any of our grants throughout the NIH, we say, we take all the data and we integrate the data and we make fantastic new tools to improve our understanding of the disease. And we have a patient cohort of X number of patients that we have thoroughly looked at with controls. If we are good, we have even a good study design. To me, a good study design is a longitudinal study design, but since this is our first grant, we do a cross-sectional only because. So that's the classic NIH grant because, right? So the problem is there that we want to achieve a systemic understanding, and usually we then say we will look at genes and proteins and metabolites, but all these parts of the system, they work on the cellular level. And what we get from patients are usually not biopsies. We usually don't get a lot of muscle biopsies or other types of organs, say brain biopsies. We get some body fluids, and most often we get blood. And if we look into blood, you see, as you see on the left-hand side, you know, that is the big interstate of transport between organs. So to try to read what is going on in the different cells under different conditions, and you only look what is happening on the interstates, it's a hard task. Specifically now, if you then want to look also at genes and where you get the genes from, maybe from erythrocytes, from white blood cells. So some other cells that are not even remotely similar to where the disease is located. And that is why this is complicated in terms of informatics, just because our study designs do not often look at the cause or where the symptoms are or the cause of diseases, but we look in blood because we are working here with patients, and that's the easiest access point. So let's have a deeper look. So this is the state of California, and we see here Sacramento is the capital of the state, so that's where the influx is of water and also politics, I guess. And then you see San Francisco, that's where the pentose phosphate pathway is, so they make a lot of money there. So then they sent it down, all the energy down to San Diego and Los Angeles, where the mitochondria reside. And as we have learned, mitochondria are really important, Hollywood, and they make more money there in terms of ATP and NADH. So this is all very important and tells us about sugars, fats, and amino acids if this was glycolysis and primary metabolism. It looks actually very similar. Now, the point is there is a lot more traffic in California. There's lots of roads and even streets in cities. And they tell us a lot more about regulation of fluxes of metabolism and more than just looking at the interstates. And these are, some of those compounds have been mentioned before by Bob Navio. For example, ceramides, sphingomyelins, they have very active roles in many diseases, but also oxylipins. They are clearly important in inflammation resolving, but also in many other physiological actions. These are usually very low abundant metabolites at nanomolar concentrations or even lower. We used to only look at hormones and cytokines, but now we need to understand that in biology, everything can be used as a signal, a systemic signal that travels across the body and then triggers responses elsewhere. So we should not just look at the very abundant primary metabolites. We should also look into novel metabolites. That includes, for example, microbial metabolites that are get into the vascular system, but also like bile acids. We now understand bile acids have lots of secondary functions and bind directly to receptors. FAFAs are involved in diabetes, and so on and so forth. So this is the reason why metabolomics is more than just fats, sugars, and amino acids. All right, and that's why at the West Coast Metabolomics Center at UC Davis, we use different assays. So on the one hand, we use so-called untargeted assays. These are both hypothesis-driven and, at the same time, hypothesis-generating. We usually do relative quantification, and we move towards absolute quantifications for these untargeted assays as well. We usually have 150 to 600 identified compounds per assay. So for example, in lipidomics today, we would have 500 identified lipids. In primary metabolism, just 150. For biogenic amines, which are TMAO, carnitines, betaines, oligopeptides, nucleosides, and similar compounds, we would have maybe 300 identified compounds. So that we overall, in metabolomics, by these assays alone, we get something like about 1,000 identified compounds in a specific tissue or blood or so. Then we also get a lot of signals where we don't know what they are. We can do statistics with them. We can annotate them by chemical classes, and as I will show you today. But we don't know the exact chemical nature of those yet. That means they might be involved in diseases like ME/CFS, but we don't know yet. And then again, we have targeted assays. Targeted assays are only hypothesis-driven. They're always given in absolute or molar concentrations. Usually these targets are 10 to 80 compounds per assay, for example, steroids or short-chain fatty acids or oxylipins. Many of those have already associated diagnostic roles. But we need to get to them in a targeted assay because they're usually very low abundant and sometimes also require very sophisticated types of cleanup tools, sample pretreatments. So what my lab really does is we try to find everything we can from a specific assay. So we produce a lot of databases and tools. Some of them I want to show. For example, MS-Dial is a tool that is everything is freely available, open access for untargeted assays, including the identification of unknowns. Oh, okay. Well, there. Ooh. By spectral matching. So this is one mass spectrum you see. And the identification works best if we have a giant database behind it. So where do we get giant databases? Well, there are some that can be purchased. For example, NIST-17 is an affordable library that we can buy from the National Institute of Standard Technologies. But today, most researchers make their spectra freely available. For example, from HMDB, that was already mentioned by Bob Navio, where they have 4,000 spectra available, GNPS in San Diego with 16,000, Mass Bank from Japan with 48,000. And from my own lab, we have added maybe 60,000 spectra that were not reported before. So we put them all together in a big mass bank for everybody to download to basically help people who have untargeted assays to identify those compounds. So when we then compare between those databases, we see that we get here about 8,000 novel compounds experimentally done that have never been shown in commercial libraries, whereas NIST-17 is clearly still the best and best also curated database that people get now. Now we say that with every single new library or database, we get novel compounds for specific compound classes. And this is how we present those compound classes. And then we see when we compare those that some of them are really better in NIST-17 and others like lipids are better or polyketides using our database. So we say that is a concerted effort and we should not disregard any of them. As I said, usually we get a lot of spectra in a profile, but many of them are unknowns, and usually we don't like to talk about those unknowns. So we recently published another paper where we said if we add more informatic tools, including the new NIST hybrid search, but also new databases like Carniblast, we get annotations across the board for every single spectrum, so that at least we know the compound class. And we do this by basically using the NIST hybrid search and asking, well, what could happen to known compounds? They could be methylated, hydroxylated, phosphorylated, acetylated, or other types of modifications can happen to molecules. And when we do a hybrid search, we move those ions around to known compounds, then we are able to annotate them. We wouldn't know exactly where the hydroxylation or the acetylation happens, but at least we have a good feeling of what type of compounds are these unknowns. And we can use them in, for example, big classes and use these classes for statistics, for enrichment statistics, so that we see in a certain disease or a certain phenotype, we have an enriched class of dysregulation, in benzenoids or in organoheterocyclic compounds beyond the known classic compounds. All right. So next thing, what we did is we said, look, if my lab is doing good work and Bob Navio's lab is doing good work, that's all fine. But then we publish, and then people have to recover those data from publications. That is incredibly hard. We need to fill databases, and we need to do this in a validated and harmonized way. We can standardize a lot of things, but we will not be able to standardize the actual machine that people will use, because in capitalism we like competition, so there will be different companies offering machines. We used here the same 155 human plasma samples and first published the biology, and then we published method validations on top of them. We repeated the same analysis 9 times with 9 different mass spectrometers from different vendors. And what you can see here on the right is principal components analyses of these treatments. This is before and after a certain intervention, and the different colors are different subjects. And then we had a longitudinal design, and we see that the overall phenotypes, the metabolic phenotypes, was always retrieved very accurately for each of those subjects, with no matter which mass spec we used. So that is one of the criticisms that have been raised in metabolomics in terms of validation. We can validate it. We can move, even with untargeted analyses, further and fill databases so that we can better address what is normal and what is a dysregulated state. The next thing is we also run often very large studies. Now in ME/CFS, the sample sets are very small, maybe 50, 100, or 200 subjects, but very, very small. In other cohort studies, we easily run 5,000 samples or 10,000 samples. And what happens then is the machine would give us errors based on the machine cleaning, or we would need to change maybe a column or buffers, or we would somehow handle the physics of the machine, and then you get a different response, and this has to be normalized. And classically, this has been normalized by locally estimated scatter plot smoothing, but as you can see here, some of that method was over-normalizing, so that we still got residual variance in the data set. That is bad because it increases the technical error, and therefore we lose statistical power. That's bad. And so we use now different methods, random forest machine learning methods that works great on large cohorts and also stabilizes, again, the idea it validates metabolomics to be used in filling databases. So what else needs to be done? So far, reviewers in NIH grants only look at, has this investigator published? Has this investigator good mass specs? Oh yeah, this is an expert lab. Now this is not good. The NIH needs to change some rules in terms of reviewing grants, because if we want to compare results apart from manuscripts, if we really want to fill in data into databases, we need to have a lot more quality controls to systemize and normalize data. In our lab, what we do, we have NIST reference standards. We have pools from the cohorts that we analyze. We have blanks and commercial pools. You can also add, and some other labs would use mixtures of external compounds, up to 1,000 compounds I have seen. So these are different ways to do it, but as you can see now, we have here 4 types of injections before we then do 10 samples. So this is important, and the point is, if these data are not uploaded, I cannot compare and contrast the data and validate data across different laboratories. So this is not required right now, and it needs to be required both for databases as well as for grant reviewing. Next time you review a grant, please look for that. Okay, so next thing is we have seen by Bob Navio the use of MetaboAnalyst. And MetaboAnalyst only looks at known compounds and known pathways. Specifically, the KEGG database that's behind it. KEGG is a database in Japan. Now, biochemical databases are incomplete. It relies on the definition of pathways. You ask 10 people, and they all have different definitions of pathways. So therefore, the statistics behind those pathway definitions is different. The MetaboAnalyst uses an inappropriate statistical design. Also, of course, the background database for what is the metabolome is not defined, and you lose many identified compounds that are not found in biochemical databases. So instead, we use the chemistry itself. We put compounds into chemical classes, for example, unsaturated triacylglycerides, and then we get very similar results in terms of the differential regulation of metabolites, of phenotypes or metabotypes. But it's not depending on a specific biochemical database, because it's only depending on did we identify the compound or not. So if we then have a final result, we need to convert this result to regular people, and that is often what we showed. So that is actually from ME/CFS patients in the beginning. So we'd say, well, we know about serotonin. It's an important neurotransmitter. And so there's a breakdown of methoxytryptamine, but also tryptophan is broken down to indole lactate. So, we see these 2 compounds. They might refer to that pathway. And that is a legitimate way to talk about data in a pathway or in a study. We can also talk about cytoplasm versus mitochondria, where we see, oh, some vitamins were increased and TGs were increased versus PCs were decreased in ME/CFS. So we saw that, and we can talk about it, but then we sometimes have confounders. We found, for example, ME/CFS patients have higher intake of vitamins, and that would easily explain the increases in pantosynate and nicotinamide. So there is this cellular view that needs to be confounded by what we know from the metadata. We can't just do the statistics. So instead, we now do the text mining to link phenotypes to metabotypes. So we now say, instead of relying just on databases, we need to rely on everything that has ever been published. Okay, NIH is very much involved in this because we use NCBI tools here. And here we first ask the question, what do we know about blood anyway? What compounds are in blood and how many? What has been reported in the last 150 years? So we downloaded 38 million abstracts and 8 million full research papers, including supplements. We looked at 4 million wiki sites and many other tools to say, well, what is known in blood? And that is what we know. So in 1 million publications, we found that metabolites were analyzed in blood. And there are 3,000 compounds that have been found more than 100 times. So these are very likely, the classic metabolome that we should look in the blood. But there's also 21,000 compounds that have been published at least once in blood. Many false positives will be in here, but not only false positives. So that gives us the first time the idea of how big the metabolome is. We then used in the first test 50 cases versus 50 controls, and we published that last year. And so this is the different types of metadata we got. But you see, we don't get a lot of history. We now understand that there's always a trigger. That could be an infection trigger, could be a stress trigger, could be many different triggers for different patients. We don't get a lot more, information. For example, the duration of upright activity that Dr. Bateman told us about. So that is not something we got here, so we can't do the statistics on it. And more importantly, what are the controls? How can we really compare these controls? So these are really important questions that we right now can't address. So we used both we used 3 of our assays, or 4 of our assays, but we also, with Ian Lipkin and Columbia, used then fecal bacteria and looked at if there's commonalities that can we integrate this? For diagnosis, for example, can we learn about it? And we then did the enrichment statistics analysis that I showed you before, where we saw that PCs were decreased, TGs were increased. So that's similar to what Bob Navio found. Increased also ceramides. These are the biofunctional lipids. And a couple of others, of course, individual compounds that were different, but not systemically across all the compounds in a certain chemical class. And then we also looked into how is that confounded with IBS, because a lot of ME/CFS patients have also IBS, so we need to be distinguishing, is it specific for ME/CFS or is it systemic also across IBS? And then we can basically refine these, and we see that it's also still the TGs and the ceramides were still different, for example. So then we can display this through chemical mapping, just because it's a little clearer than a table. I mean, it's the same information as a table, but we see now clearly that the lipid regulation is different, including LPCs. So these are partial degradation of lipids in terms of getting at one acyl group. TGs higher. So overall, that might indicate a problem in lipase activities. But which lipases? There are many lipases. We don't know which ones right now. We see the ceramides and sphingomyelins here dysregulated here. We see also changes in acylcarnitines. Acylcarnitines are also a good marker for mitochondrial activity in terms of overload of mitochondrial activity, and here we see that they are downregulated. So a lot of this can be already discerned from such kinds of overviews. When we now integrate bacterial information from stool samples, we see that in the integration, we get better recognition than by either of these omics tools alone. So the metagenomic and the metabolomic tools together were better able to distinguish the controls, which are in yellow, from the ME/CFS patients in red, no matter what type of lens or bioinformatic view you would take on this data. So we, since we published that, we continue that in CSF fluids because it's closer to the disease. We see clear differences depending on which kinds of controls we use. We haven't published that yet, but I think it's ongoing. And so you will learn more about that, and my time is kind of up. So I want to tell my last 2 minutes or so about how I see it, and Bob Navio also thankfully did a little bit on that. I really think that metabolomics has to become more standardized so that we can better communicate across. For that, we need kits of internal standards to do absolute quantifications, just like Bob Navio said. We need single software, and my lab does this through cloud computing that is able to take in any data from different laboratories through the same type of data processing. We need to be faster. So we opened a secondary lab in Sacramento to offer 2 weeks turnaround time, and eventually, at least for UC Davis, 24-hour turnaround time so that we can actually do this cheap. We also need to be cheaper. So we lease instruments, we go for robotization. So that's all data acquisition. But in terms of linking to population health, we need a much bigger databases to tell, what is a normal population? What do we see normally, not only in these few controls that we picked here randomly, but really across the population? And we don't know that yet. And we need to link to text mining instead of genetic and genomic databases, because the best of what research has done over the last 50 years is found in papers. We need to get it out of the papers in order to do complex interpretations. So overall, we have, I've shown you some of the tools that we have developed and how they can be used. Some initial lessons we learned from plasma differences, similarities and differences to what was published before. And we see that we can integrate data through multi-omics, but we also see that it's not exactly easy to do. But there are tools from literature text mining, metagenomics, and other types of multi-omics tools that we can use to better interpret and also diagnose subjects. So overall, I'd like to thank you for your attention, and specifically I'd like to thank Professor Ian Lipkin and the ME/CFS consortium that he leads, and the NIH for funding. Thank you for your attention.